So first of all, welcome Visionary Sisters. We're delighted to see you, and a very warm welcome to all of our special guests from around the country, particularly those guests that may be attending a Visionary Women's Salon for the very first time. We're delighted to have you here, and we hope this is the beginning of many more. My name is Shelley Reid. I am president of Visionary Women. And many of us, we're all facing the challenges of this new world that we live in. And Visionary Women has, I would like to boast, successfully been able to navigate this new world. Our goal during the last 7 months has been to remain connected to our membership, to maintain engagement with our membership, and to continue to deliver to our members and our guests the best of the best, first-class, A-plus programming that we've been able to deliver to you in the past 3.5 years when we were all able to meet in person. And today we are fortunate to bring you yet another best of the best in terms of female thought leaders, trailblazers, and female luminaries. So get ready. I think this is going to be a really wonderful hour together. This is an example. The Women on the Frontier of Science is an example of Visionary Women's commitment to being ahead of the curve. And to be just a little boastful again, we reached out to Jennifer and ICI before she received the Nobel Prize in chemistry. And we're very fortunate today to have here two remarkable women, both in the world of biomedical research. Johnese Spisso is the president of UCLA Health, the CEO of UCLA Hospital Systems, and Associate Vice Chancellor of UCLA Health Sciences. She is a nationally recognized healthcare academic, and in addition to all of that, she oversees all operations for UCLA's many hospitals and clinics, as well as all of the outreach strategy for UCLA healthcare system. She is an active community philanthropist, the recipient of numerous awards, and most recently has been named Modern Healthcare's top 25 most influential business leaders in 2019. Prior to that, she was awarded the LA Business Journal 500 most influential leaders in Los Angeles. Welcome, Johnese. Our guest, our second guest, who works for the Innovative Economics Institute Governance Board is, excuse me, I lost my piece of paper here, is Megan Hochstrasser. And Megan is a graduate of Brown University, as well as received a PhD at UCLA, at UC Berkeley. And she is really quite fabulous. She's young and vibrant and working on the cutting edge of all sorts of biomedical research involved in genetics and being able to predetermine biomedical outcomes and define diseases. I would like to introduce both of these incredibly talented ladies. And if you don't mind, I'd like to be the first to ask you a question, which is, you're both trailblazing women in the field of science. And I'm wondering if you would share your career journey with us in terms of who inspired you, how did you get interested in the healthcare and research and sciences world, particularly in science when it's been so typically male dominated?
Well, Megan, do you want to go first, or I can jump in?
Sure, I'll go first.
Great, thank you.
First off, thanks so much for having me. I'm excited to meet everyone and chat with you today. How did I get into science? How did I get inspired? I think for me, my dad is actually a scientist, and even though he never like pushed science on me very strongly or told me what I should do in my life, I think having that influence changed the way I saw the world just from day one. And because he has his own lab and everything, I would always go to lab parties and things as a child, and I would see all kinds of people in his lab. Men and women alike. You know, I never really saw it as a male dominated thing from my child point of view. So I think I always just kind of saw science as a possible career option. And I was always just super fascinated with nature, like digging stuff up in my yard and collecting rocks and everything. I just found it super fascinating. And as I got older, I just continued to be interested and got more and more interested specifically in doing actual research. And once I started on that path, I had a lot of really powerful female mentors in my life. So even in undergrad, when I started doing research, I worked with a principal investigator of a lab named Elizabeth Ulu at Yale, who personally mentored me, which is very peculiar actually for someone who runs a lab to personally mentor an undergrad. So I learned a ton from her. And then in grad or later in undergrad, I worked longer term with Tricia Serio, another amazing female biologist. And then in grad school, I kind of just stumbled into Jennifer Doudna's lab because I was really interested in CRISPR. This was before it was used as a gene editing tool, when it was just this kind of esoteric topic. And Jennifer is you know, she just won the Nobel Prize. So she's a pretty inspiring person to be around, and she's really accomplished. So I've just been kind of surrounded by strong, intelligent women my entire life.
Well, thank you. And, you know, I have spent my career in academic medicine in leadership roles throughout the organization. I actually started my career as a registered nurse working in critical care, emergency, and trauma, and then came up through nursing leadership and then hospital leadership, always in academic health systems, whereby our focus is not only the clinical care, but the training of the next generation of health professionals and that leading edge science and discovery that really helps us lead to greater outcomes for patients, better cures for diseases. So I always had an interest in working in academics. I come from a family of healthcare professionals. Most inspiring to me was definitely my mother, who was a principal, a teacher in a school district, and became one of the first female principals, really at a time when very few women worked, let alone were in leadership positions. So I was always raised with, you know, the thought that yes, as a woman, you can do any type of job that you want to do. So I definitely had good role models. And throughout my career, I definitely have had amazing role models who have supported me and I've learned from and have really supported the development of everyone, especially taking a special interest in women leaders. I'm the first woman to have this position at UCLA, so definitely was thrilled for that opportunity as well, and really try to spend a lot of my time mentoring other women in healthcare leadership positions. And I'm thrilled to be here with you all today, and just delighted to have the opportunity to do the interview with such an amazing scientist like Megan.
Thank you. Well, I think that we'll turn the baton over to you, Johnese, and I think the end result of both of those answers is that role modeling is critically important. And now, of course, Johnese, as you being the first female appointee in your position, you are creating the new role model. So thank you very much.
Thank you. And thank you all for devoting your time this afternoon. We've got some exciting questions and a chance at the end to take some of your questions as well. But we first wanted to kind of open this up in this amazing field that we're hearing about with CRISPR. Megan, wanted to ask you, how does this cutting edge research in the field of CRISPR, how is that being used now to treat diseases? And can you give us a few examples? And what do you feel the impact of this is?
Yeah, great question. So CRISPR, just a little bit of background, is essentially a tool that scientists can use to rewrite DNA. So we can change a DNA sequence from something that might be causing harm, maybe you have a mutation that's causing disease, to something healthy. And it was really developed in 2012, 2013, and just in the past 7 years since then, it's already starting clinical trials. So it's being tested in humans to potentially treat a variety of conditions. And I think beyond or before even the clinical impact that it's going to have, I think it's important to note that it's actually really changed how we're able to even do research at all. So way before we get to the point of treating someone for a genetic disease or for cancer or something, we're able to test with either that patient's cells in the lab or other cells, animal models, all kinds of different organisms. We can manipulate their DNA and test things before we do anything in a human. So big changes in the research landscape. CRISPR's become a tool that is used in basically every biology research lab around the world. And in terms of clinical development, there are a couple different ways CRISPR is used. So I guess the main thing that is kind of the most exciting right now is trying to use CRISPR for genetic disease. So genetic diseases, because they're caused by a mutation in DNA, there aren't really good ways to treat them without changing the DNA itself. So for most genetic conditions, the potential treatments are kind of treating symptoms more so than curing the disease or trying to fix the underlying cause. So with CRISPR, we're actually a lot closer to being able to correct those mutations. And I think the example that is sort of furthest along right now on that front is using CRISPR for blood disorders like beta thalassemia and sickle cell disease. So NPR has actually been following this one sickle patient in America named Victoria Gray, who I wonder if she knows how many times a day scientists are saying her name, because we're all so thrilled to see that CRISPR has been working for her. So she seems functionally cured of sickle cell disease. And this is someone who had very severe symptoms before this treatment. And it seems like the effects are lasting. So we still need to monitor her and other patients in this trial to make sure that there aren't side effects or anything that come up later. But, to me, having been in this lab where CRISPR was developed and not thinking it would really go anywhere, because honestly, I'm a skeptic at heart, it's really just incredible to see someone's actually already been cured of a genetic disease in less than 10 years since this thing was even developed.
Wow, that's amazing. I was noticing here at UCLA, one of our researchers, Leo Krugliak, from our David Geffen School of Medicine at UCLA, they have worked on tweaking the technology, enabling them to monitor the outcomes of tens of thousands of gene edits in the time that it usually takes to analyze a few. And that is really using the powerful gene editing tool that CRISPR has revolutionized in this research. And so it seems like there is even additional progress being made to help with the cost as well. So can you describe a little bit about that, the cost, and how the importance and relationship of that is with using this technology?
Absolutely. Yeah, I think that one of the wild things about the gene editing field is that we're constantly improving the technology itself while also using it in clinical settings, in agriculture, all over the place. And so it sometimes feels a little bit like building the car while you're driving it. So there's often really big advancements happening at the same time as applications. And I think cost is really a huge issue actually for clinical applications. In other senses, like in agriculture, I think CRISPR will bring some costs down. In the lab, it's actually much cheaper than other gene editing approaches and other tools. But for medical treatments, I think it's going to be very, very expensive. And this is something we're really constantly talking about at our institute, because Jennifer founded our institute basically to make sure CRISPR actually solved problems in the real world and didn't just stay in the lab. And one of the hurdles that we have to get past to make sure we really have the impact that we want and that we really benefit patients is making it accessible. So right now, similar approaches, not CRISPR, but other therapies that involve manipulating DNA have run hundreds of thousands to millions of dollars per treatment. And it's not clear that every insurance company is going to pay for this. As you know, tons of people don't even have insurance. And so that's a really high high cost for a treatment, even though it should, in theory, permanently correct a mutation and won't require additional doses. But we want to make sure that we're trying to do everything we can to bring that cost down to make sure this actually benefits people. So at IGI, we're trying to think of what we can actually do to change the technology itself to make it more cost effective, to remove barriers that are adding cost to the process. So I think we'll probably see maybe the first handful of CRISPR therapies approved being really, really costly, but the community is very aware of this and we care about it a lot. So I think steps are being taken that will bring the cost down over time, hopefully.
Yes, that's great. And we can share some of the links to these articles as well. The one thing about this UCLA study that was reported in Nature Genetics, it really allowed them to really edit so many genes at once and really do this on a large scale area, which, as you said, is so important.
I don't want to interrupt your flow, but I think a study like that is really important because while some of the first things we're going to treat are things like sickle cell disease, which is caused by one mutation, to really treat the whole spectrum of genetic diseases, we have to be able to make multiple edits at multiple places in DNA. So there are a lot of more complex diseases that we really can't even begin to treat until technologies like what you're mentioning are advanced.
Yes, thank you. The next question, how is CRISPR being used outside of medicine?
Sure. So I think I touched on it being used very widely in basic research. I think one example that's really interesting to me is there are labs that use CRISPR to edit butterfly DNA to learn how butterfly wing patterns develop. And they can change the pattern of the wing and the coloration by changing the butterfly's DNA. So that's not going to turn into a medical treatment right now, but it really is helping us understand nature and simple things about how what's encoded in an organism's DNA causes traits in the organism. And then I think the other big area that gets talked about a little bit less often is agriculture. So traditional plant breeding and other approaches to modify plant DNA are really costly, time-consuming, and technically challenging. So the plant world and the livestock editing world are really excited about CRISPR because it makes a lot of their work a lot faster. And it's much, much more specific and precise. So I think if you've heard of like the previous GMO technology that kind of often gets a bad rap, that technology involves inserting a piece of DNA somewhere into the genome of an organism. So you can't really control where, you're just throwing in like maybe a pesticide resistance gene from an organism somewhere. With CRISPR, what we can actually do is make small little mutations or changes. So we can add something, we can delete a little portion, we can make a single genetic letter mutation. So we can do a lot more fine-tuned control, which opens up a lot of doors and is a lot more precise, as I said. So I think the other angle on that is that because this is a less expensive technology, a lot of smaller companies are able to get into this agricultural development game. So I think one of the criticisms of that field is that it's really dominated by these huge agricultural giant companies that have a lot of issues with how they've dealt with their consumers. But I'm very hopeful that smaller companies can start to make their own products and that maybe even academic labs can quickly get some of their own products out into the world to help farmers trying to keep their crops going or to potentially combat things like climate change. We can try to make drought resistant rice or submergence tolerant rice, all kinds of different crop properties that could help combat climate change.
So really exciting to see the use of this really in medicine and also in so many other important fields. Another, I think, important and exciting opportunity with this is around the treatment in cancer. And just in May of this past year, 2020, we had researchers at our Broad Center of Regenerative Medicine at UCLA and our Johnson Cancer Center that figured out a way to essentially genetically edit cells that researchers in the past have had a lot of difficulty with. This was in the mice model. And the real impact is to really take those cells, edit them from the patient's blood and put them back in to really function as an immunotherapy to fight cancer. So while this was right now in a mouse model, there's definitely a lot more going on to use this to do those genetic edits in order to treat significant diseases. And I know we get a lot of interest from the public on what's happening around that as well. So many, many positive things about this, but we also all get questions about some concerns and dangers. So Megan, wanted to ask you, from your perspective as a leading scientist, what are the dangers of genome editing and what are the possible off-target effects to society?
Sure. So there are a lot of complicated ways to answer that question. I think when you have a really powerful tool, there are good ways to use it and there are bad ways to use it. And I try to make sure people don't vilify the technology itself. It's really the people using it who have to decide what is a good way to use this, what is a responsible way to approach this. I think, let's see, inherently, because the way the technology works is by finding a place in DNA and making a cut and a change, you can have inaccuracies like the off targets you mentioned, where we cut in the wrong place by accident. I think that has actually, things have really improved on that front. Like we kind of very quickly identified that as an issue and lots of work went into improving the system, ensuring minimal off-target effects, making sure the on-target cuts and edits are what we want. So I think things have improved a lot in that sense. I'm not super concerned about off-targets clinically. I think one thing we're still kind of trying to figure out is the on-target cutting. So sometimes when we make an edit or we make a cut, we don't just get the little small change in the DNA that we want. We might have like a whole chromosomal rearrangement or a big deletion or some big change that we didn't anticipate. And so I think that's something that we're still trying to figure out and make sure that it works okay and is safe for people. And I think a lot of this, you can't really test everything in a mouse. You can do that, but then at some point you have to switch over to a human. So I think people who are advancing clinical trials in humans are being really, really careful as they can be for the most part. We can talk about the Chinese embryo editing after this. But I think most people are trying to be really cautious and anticipate all of these possible changes and effects ahead of time and kind of set up ways to prevent them. So there's some kind of technological issues. To me, the bigger issues are the ethical issues and the moral issues of how should this actually be used? Should we be able to edit human beings and change things that are aesthetic or not, not related to disease? Will it be regulated properly? There's a lot of ethical issues beyond just technological issues.
Really important ethical issues that have so many other implications and unintended consequences. And related to that, and certainly what you've just mentioned, we have heard that there are Chinese scientists creating designer babies, if you will. How is this different from cloning? And again, from your perspective, what are those ethical implications with that?
Yeah, so CRISPR is a separate technology from cloning. I think there's sometimes a lot of confusion when you're manipulating embryos or DNA. What exactly are we talking about? Cloning is where you're trying to take an organism, take its whole genome, all of its DNA, and put that into a new organism so that you create basically a copy of that organism. And I think people should know that you're never going to really be able to copy a person or or an organism. Like I've heard that Barbara Streisand, for example, has had her dogs cloned because she really liked her original dogs. But I think it's important to keep in mind that that new dog might look like the original, but there's no reason to think it would have the same personality or the same qualities that she liked about the first one. So cloning is a different thing from CRISPR, and it's not allowed in humans. I'm not aware of anyone ever trying to do it in humans. I think that's like a big no-go. Everyone knows it's sort of out of bounds. CRISPR is making changes to DNA in an existing person or in an embryo, and that's what happened in China. So a couple years ago, this Chinese scientist that people didn't really know about beforehand kind of shocked the world by announcing that 2 twin girls had been born that he edited when they were just embryos in his lab. And there's so many reasons why he did a terrible job at this. So he really botched it scientifically. But the bigger issue is really like the ethical implications of this. And he's actually, I believe, in jail. So he was charged for the crime because he forged permissions and all sorts of things. But I think it really reignited this conversation that we've been having in the scientific community, along with various regulators and bodies like the National Academies of Science, the United Nations, WHO, all these groups. And we're trying to come up with good guidelines to make sure that that doesn't happen again. And that if people do try to edit germline cells like embryos or sperm or eggs to try to change an eventual person, that it's done in a really safe manner and an ethical one. So I think the one of the bigger issues that comes up there is if this were to become really easy, like you're doing IVF and your doctor says, oh, we can also offer you this array of edits to this genome. I don't know what we're going to allow on that list. So I think some people would say it's okay to try to prevent a disease. Like if we've sequenced an egg from someone and we know that they're going to carry some really devastating disease, some people think that's okay. Others don't even think that's okay. But I think one of the issues is if we could make these sort of borderline enhancements to people, that gets really dicey. So I don't know if you've seen the movie Gattaca. That's like the example we always go to. I think a lot of people are fearful of having these genetic haves and have-nots and further exacerbating systemic inequalities in our society by having some people who can access these enhancements and some who can't. So there's a lot to be debated there. The only point I would make on that is that right now, we really can't do many enhancements that anyone would care about. So we can't like make a really intelligent baby or a really beautiful baby. We don't have the understanding of genes to do that. So right now it's kind of limited what could even be done, but now is also the time to be having the conversation.
Yes, yeah, really good points. All right, let's move on to some of our current events. And certainly the worldwide pandemic has been front and center for all of us for many, many months. What are your thoughts about how CRISPR could help in developing tools to combat coronaviruses or pandemics like COVID-19? And is it possible that it could modify genes in the coronavirus to really help ease the pandemic?
Yes, so CRISPR is definitely playing a role in the scientific community's response to coronavirus. There are a handful of people who are trying to pursue directly using CRISPR to like cut the viral RNA to stop it from infecting cells. I think that's a little bit farther off. I don't anticipate that having a big impact during this pandemic anyway. But I think the major role that CRISPR is playing is in diagnostics. So this is a kind of a different use of the same set of CRISPR proteins that are used for gene editing. And instead of finding a place in DNA and cutting it and making it change, we can use the similar proteins to find coronavirus RNA, the coronavirus genome, and tell us that it's there by cutting a little fluorescent flare thing that we can detect. So it's thought that CRISPR-based diagnostics could be very, very sensitive and fast and inexpensive. So there's a couple different companies and a lot of academic labs, including Jennifer's lab and others at our institute, that are trying to develop these technologies really, really quickly now that it's, you know, crisis mode, crisis time, to make sure that they can have an impact during the current pandemic. So I think there's been a lot of work done in the lab. It's still being translated into something that will reach real people. So these are not tests that are really available yet outside of a lab setting. But the hope is that we could potentially make something similar to like a pregnancy test that you can just use at home, put a sample on it, and see whether you have the virus. And that's all in development right now.
Great, that's really exciting. We have two more questions and then we'll get to the questions from all of you so that we have enough time to cover those. The next question we had is, how do healthcare professionals and hospitals deal with the rollout of a vaccine for something like COVID-19? So I think I can address that because we've been actively planning that for several months. I think many of you know there are many active trials going on right now for a COVID-19 vaccine. Here at UCLA Health, we are a participant in 3 of those vaccine trials. So every day we get additional information and updates. We've also been working and planning with both the state and federal government once a vaccine is available to assure that we can get sufficient quantity of that vaccine. So we have been, for the past several months, filling out different questionnaires and information that goes in. The same way we actually had to do that when the new drug remdesivir was available to treat COVID-19, and we had it on a clinical trial here. It rapidly came off trial, and then we had to work nationwide to make sure all hospitals could get enough of that. So we actually feel really good about our ability to deliver mass vaccination. We do that routinely every year with the flu vaccine. So right now we're in the midst of really getting flu vaccines to all 200 clinics that we operate through Los Angeles and throughout Southern California. And so the motto is in place. The big thing will be, when is that vaccine available? And is that vaccine a 1-dose, 2-dose, 3-dose vaccine? And then again, how do we make sure that we're able to get the quantities we need? So there is that planning in place and in progress right now. And then the last question that we had was, we get a lot of questions of, when do we think we will be going back to pre-COVID days, and are there phases? So I'll give you a little bit of my perspective from the healthcare system that's been dealing with COVID since actually even before the outbreak started in LA. We had been working with our teams on our international program to follow what was going on in China and to get ready and prepare. So our work actually started in January and February of last year, and we began being the screening site at UCLA Health for everyone doing international travel with CDC screeners based at LAX and looking for any symptoms. So we definitely have been in pandemic mode for many, many months. The one good thing I'll tell you is we are really pleased to see the very low numbers of hospitalization that we have right now. So we had at our peak within Los Angeles, for example, we were averaging about 2,200 patients a day in all the LA hospitals with COVID. At our peak at UCLA Health, we were running about 60 to 70 patients a day that required hospitalization. Now in Los Angeles this week, we're averaging about 750 patients in all the hospitals that have COVID-19. Here at UCLA, we have been running anywhere between 5 to 10 patients. So it's down really low. So even though we're seeing a lot more transmission in the community, it appears that that's in a more younger, healthy population. People aren't getting as sick. So going back to normal, we have had some ease of the restrictions here in California and Los Angeles. We still haven't brought our students back to campus. As far as going back to normal to when we can have large events, things like that, my personal feeling on that is that won't be until we have a vaccine. And again, there is a lot that's been in the press about, well, why don't we just open things up and get the herd immunity? I think you've heard what the experts feel about that. There would be a lot of deaths in between until we got to those numbers. So we are still definitely advocating, caution and following the guidelines coming out by the CDC regarding reopening. And Megan, I'll turn it over to you to see if you have any thoughts on that as well.
Sure. I mean, I agree with everything that you said. I think that's not really my area of expertise, but we've gotten into, of course, like everyone has kind of into coronavirus research at our institute as well. So I think I agree with you on that front. I had one question for you actually on that.
Oh, yes.
How do you go about, assuming there are multiple viable vaccine candidates, how do you go about choosing a winner and what you're going to offer? Or do you offer multiple?
That's what we have been talking about, because right now, even on the trial, we offer 3 different ones. So what our researchers will tell you is we will probably advocate with whichever one looks the best, has the least amount of side effects and things, and something that we can really have in a more of a mass produced quantity, quickly, because obviously everyone is going to want that soon. So even though we're doing 3 trials here, we're actively following more than 9 trials that are being completed worldwide. So
Gotcha.
Yes. The other thing that I'll say is one of the things we definitely have learned over the past many months is better ways to treat patients with COVID-19, even patients who are severely ill with it. During the course of the hospitalizations that we've had at UCLA, and we've had over 700 patients that have been hospitalized, you know, I'm pleased to say that the mortality rate has been relatively lower. We've had right around 50 patients who have died from COVID-19. The majority of those patients were early on, and they were elderly with a lot of pre-existing disease. But we've had many patients who were very sick for weeks in the ICU, on ventilators, even on extracorporeal membrane oxygenators, who we were able to discharge from the hospital, and they're you know, being followed as outpatients now. So there definitely has been advances made in the treatment of it, and that information doesn't always get out there, you know, to the public, but that there's been a lot of work, and we've been collaborating nationally with other leading centers, and everyone sharing the expertise so that we all can be really providing the best care to these patients. Great. So with that, we're going to open it up now for all of your questions. And thank you, those of you who put the questions in the chat. We're going to be taking as many of those as we can. But I wanted to start with our first question. And our first question comes from Melanie Fine, a Forbes contributor and founder of Rocket Girls, an initiative to support girls in STEM programs. And the question is, from Melanie, the research is clear. Women leave science careers at every point in the pipeline, and now, under our current leadership, scientists are ignored or maligned. My company, Rocket Girls, works with high school girls to help ignite them in a lasting passion in science. What do you think is the #1 thing we should be doing as a country to bring more girls into STEM fields? And are there enough jobs for them once they get there? Well, first of all, Melanie, thank you for your work and your leadership at Rocket Girls. This really is amazing. And I'll turn it over to Megan to get her thoughts on what she would say is the #1 thing we should be doing.
Yeah, that's a great, difficult question. I'm glad that there are groups like Rocket Girls for sure out there trying to do this. I guess the more I think about this, the more I'm kind of convinced that it's all about supporting people once they show some kind of interest, and trying to find ways to show people that they belong. So not even just women, but scientists of color or queer scientists, and especially people who hold multiple marginalized identities. I think there's a lot of work to be done to show people that they're appreciated, that they're valued, and that they're really wanted in these fields. So I think some of that can be accomplished through better representation. So changing the stereotype of the like zany haired white male scientist, and showing that scientists are actually really diverse, all kinds of people, all kinds of personalities. Science is not just for people that are like study happy bookworms or something. It's really all kinds of people that do all sorts of hobbies and activities. So I think just showing people what it can be and finding ways. I don't have a great solution for how to support people once they're in STEM, but I think valuing diversity, I guess, throughout, and encouraging people, hiring and advancing people's careers based on what they've done to advance inclusion and bring in new types of scientists to their field, not just valuing papers and the things that we kind of traditionally look for as success markers in science. I think it kind of changes. It's going to require a frame shift, or a shift in the way we perceive science and what is success in science. So I don't know if that's a great answer, but there's so many things we can be doing and need to be doing.
I think that's it. And I would also add on in relation to jobs. I know one of the things that we're doing at UCLA Health is we have an innovation program, and we fund innovation fellowships for people who are doing any type of research in the healthcare, in the clinical setting. And we really try to provide those opportunities, and we have really what we call a startup in a box. So it's not just them, we surround them with some of our other resources, so that as young researchers, they can get a little bit more support and mentorship. And Melanie, I'd love to connect our innovation chief with you to maybe partner with Rocket Girls, because we'd love to really start working with women at younger ages. So we'll connect with you on that. So thank you. All right, let's move on into the chat. And I see a great question from Barbara. Hi, Barbara Lazara. The question is, and this is a good one for you, Megan, how do we respond to the controversy around CRISPR when there's so many important and groundbreaking uses and future hopes for some of the things we've mentioned, sickle cell and so many other genetic syndromes? What are your thoughts on that?
Yeah, I think my approach is to make it clear that CRISPR is not one thing. Like if we do one application of CRISPR, we don't have to do another. So the controversial things that are done, like, these babies that were edited, that doesn't have to happen. We can still use this tool for really good things that we agree are going to add value to society, and we don't have to do the controversial things. So it's not like an evil tool. It's just, we have to choose together how to use it. And one thing to note is, like, CRISPR scientists are constantly talking about this. We're always talking about what's the ethical way of using this technology. And there are constantly calls for more public engagement, because scientists don't want to be the only ones making decisions about how a technology is used. We really want the whole world to know this exists and to weigh in and guide a responsible use. So I think we really welcome hearing from people what they think, and how you think it should be used. So I always just try to emphasize that people have power in this sort of context. It's not all about what the scientists think, and feel free to get involved, and also to value like anyone's opinion. You could think it's great to do editing in an embryo and enhance someone to try to make them more intelligent. That's okay to have that viewpoint. I don't reject that. You can think whatever you want. But to me, it's most important to be informed and to try to participate in the conversation.
Yes. I think as well, as academic institutions, we always try to use advocacy groups so that we're hearing those voices from the community as well. And really then, advocating for the right type of legislation or regulation around some of these things so that we can be really best representing the interests of the public. But it does definitely take those collective opinions and voices coming in, and definitely, helping set some of the guidelines and policies around that. All right, the next question is.
I was just going to say, someone followed up in the chat. I just saw to say, how do we give that kind of feedback to scientists? And I would say it's kind of what you just said, trying to influence regulation and policy, but also we all have events. Like I'm running an event 2 days from now that's all about sickle cell and the ethics of how to apply CRISPR in that context. So I would say attend events, try to talk to scientists if you can. There are a lot of events out there that are really open to the public where scientists are trying to talk to the public, but you don't always know how to reach people. And talk to your family, write blog posts, talk to reporters, talk to anyone. I think it's just talking about it and trying to get other people to know what's going on and just joining in.
Great. Another question. There seems to be controversy around genetically altered crops. Do you have time? Can you explain that?
Yeah, sure. So this is when I entered the CRISPR field, I was coming into it with this whole long history of the GMO debate. And it's been really interesting to try to get up to speed on everything that's happened there, and then try to figure out how CRISPR fits into that picture. I would say that GMOs honestly have a kind of an undeserved bad reputation in a lot of ways. I think there are a lot of people that are really, really gung-ho against it for a variety of reasons. But most people that I talk to anyway don't really know a lot about it. They just kind of have heard that GMOs are bad, so they avoid them. I think the reason people kind of hate them or have come to hate them is because the way they've been rolled out by companies is not so great. So I think people just automatically don't like a big company. Some of the companies involved have done kind of shady things in the way they've dealt with consumers. And I think that those bad feelings towards the companies have kind of rubbed off on the science itself. And science-wise, there's really no danger inherent to a GMO. Like just putting a gene from one organism into another to have a benefit is not bad. I think you could say, oh, I don't want, this is nature, I don't want to mess with it. But I think you have to acknowledge that when you're breeding crops, you're just basically, they're mixing their DNA together and you're getting some DNA in the product plant that you're not controlling. So to me, using CRISPR or even GMO technology to change DNA is way more precise than just breeding and breeding and breeding plants, shaking up their DNA like it's a boggle square and hoping you get some good outcome. So I would say, as a scientist, GMOs are not inherently unsafe. I think that the only thing that's really going to change public opinion on this is if we get GMO crops that are actually appealing to the public for some reason. So far, the companies that make them are making them disease or pesticide resistant so that they can sell the pesticide that you need to use with them. And no one, I don't care if my tomato was helpful, was better for the farmer's experience in growing it or something. I'm just going to get the tomato that looks good when I go to the store. So I think if academics or companies could make products that are interesting to people, like an apple that doesn't turn brown right after you cut into it, or something that has a consumer benefit, maybe people would see things differently. But yeah, maybe CRISPR is another chance too, because it is a different technology and it is a lot more precise.
Thank you. Another question, this one from Allison Gingold. What are the effects of altering the DNA in one sequence, and how does that potentially affect a different part of the genome?
Yeah, that's a great question. I think there are a lot of cases, so a lot of the initial work that people are doing to use CRISPR for therapeutic benefit is in cases like sickle cell, where we really understand this one mutation that causes the disease. We know what that's going to change when we change it. We know a lot about it. So that's kind of where people are working first, but we don't totally understand the genome. So there's all kinds of genes, they're regulated in different ways, they interact in unexpected ways, and we don't know that much about the genome as a whole. So I think there are definitely possibilities where we could try to change one sequence in DNA and we would end up having an impact that we didn't expect on the cell or the organism. So I think CRISPR is actually a really good way to learn about the genome, because we can just take cells in the lab and make these manipulations and see what happens. So I think CRISPR is really already helping us understand how genes control a cell's function or an organism's trait already, and we're just going to learn more and more about that.
And another question, this one from Kathy Baker. Are there gene editing applications for the treatment of blood cancers besides immunotherapy?
That's a good question. I don't think so. So most of the applications, most applications of CRISPR are really for immunotherapy. I have heard of a few people that are trying to do direct targeting of cancer cells using CRISPR. So trying to deliver the CRISPR only to cancer cells, or to only cut the DNA in cancer cells to try to kill them somehow. But I don't think any of that work is close to being used in real life for patients. Not yet.
Thank you. Another question, it said, looks like your lab is on the cutting edge with other labs for a vaccine. How will you manage the bias in all media globally to send out the correct message if a vaccine is developed, so that the population responds well and conspiracy theories don't dominate? Wow, that's been so important, hasn't it, during this entire pandemic, to get the right information out. We've done so many communications and other things to try to get factual information out from the healthcare and the research side. But there is a tendency as well for many different conflating opinions to kind of distort messages. Definitely with the vaccine, when we do have one and when it is ready, we know that we will have to develop frequently asked questions, talking points. We've already talked about on our website having a list of FAQs with answers, having the ability for patients to get more information. And I think we'll be working very closely with the Centers for Disease Control here in Los Angeles, LA County Department of Public Health, so that we can have the right information out there for patients as well. But that's going to definitely take a statewide and a nationwide effort to do that. The next question is, do we anticipate in-person events to start again in spring and summer of 2021? You know, I think that's what everyone is saying. That's probably the soonest. But again, I think it is going to be dependent on a vaccine. It's going to be dependent on how well we get through what is our normal flu season to really see the effects. But I would say that's, for planning purposes, that's probably, in my opinion, a good date. I don't know, Megan, if you have any other thoughts on that.
No, I know I'm working from home until
Yes.
spring at least.
The other question that came in for us, it said, this is a personal question, but will Megan and I be vaccinating against COVID when it becomes available? So from my perspective, for me personally, since I am in the healthcare setting, I'm here in the hospital and in our clinics every day, yes, I probably will get vaccinated if there is a vaccine. Right now, you know, we have been, for many, many months, we do a screening survey every day before we come into work, so that we have to take a self-assessment to verify that we don't have any of the symptoms for COVID. We do temperature screening at each of our sites. We've actually also, here at UCLA, we have just developed a very rapid saliva test, and we'll be rolling that out to when the student population comes back. But the goal as well would be we could have even a rapid saliva test to test all of our employees. But similar to the flu vaccine, we just, this year was our first year to make the flu vaccine mandatory for all healthcare providers. In the past, we've encouraged it strongly, but this year, in order to work at UCLA Health, unless you have a medical condition that excludes you from getting a vaccine, you do have to be vaccinated to be providing patient care. So in that context, I believe for the COVID vaccine, then yes, I would. And Megan, I don't know what your thoughts are for you personally.
Yeah, yes, definitely. I definitely will. I just got my flu shot 2 days ago, and I will definitely get a COVID vaccine when I can. Strong yes.
Yes, yes. And any of you who need flu shots, you can get them in any of our 200 clinic locations. We have plenty of the flu vaccine this year, so definitely are encouraging that as well. All right, with that, I think we are just at 3 minutes before 4:30. So Shelley, we'll turn it back to you to close the program, and any questions that we missed, we will try to get those answers back to you in the chat.
Well, thank you to Johnese and to Megan. Both of you are true innovators, and this has been a real opportunity for us to learn about CRISPR and the tools of CRISPR technology. I dare say that as a result or as a consequence of the COVID pandemic, all of us are much more attentive and supportive of the need and the value and the hard work of medical research. And Megan, thank you for acknowledging the bioethical concerns of DNA and genetic editing. It's a big subject, and it's something that goes hand in hand. And I think you saw that in the list of some of the questions in the chat. And Johnese, thank you very much for updating us on what's going on locally and for all of the good work that you're doing at UCLA. You both represent the future, and really from all of us at Visionary Women, thank you very much for taking the time to be with us today. This has been a really marvelous, marvelous program, and thank you very much.
Thank you all.
Thanks. Thanks for having me.
Thank you.
Goodbye, Visionaries. We will see you next time. Thank you for everyone who joined us. We had a marvelous turnout, and if you would like any more information about Visionary Women, please go to visionarywomen.com. We'd love to see you. We'd love to have you join and participate with us. Thank you.